Chapter 5 is going to focus on macromolecules, carbohydrates, lipids, nucleic acids, and proteins. In general, your macromolecules are polymers, and they are built by forming covalent bonds with monomers. All living things are made of these four types of macromolecules. The macromolecules themselves, again, are formed by combining atoms together via covalent bonds. You will see that they have very different structures and they have very different functions. So just in general, polymers are going to be large molecules. They can be long, they can be branched, but they are combinations of monomers that have been connected together. 3 of the 4 macromolecules are going to be polymers. All but lipids are polymers. In general, when we are forming polymers, we do that by removing a water molecule. We take a hydrogen from one unit and we take a hydroxyl group from the other unit, and the bond that forms between the two units results in the polymer forming. When hydrolysis takes place, when you add water to a polymer, you actually break down the polymer into smaller subunits. So it's the reverse of that dehydration synthesis that results in the polymer forming. The first macromolecule we're going to talk about are carbohydrates. These are more commonly known as your sugars. You can have monosaccharides, you can have disaccharides, and you can have polysaccharides. Your monosaccharides are going to be your simplest carbohydrates. Examples include your glucose, your fructose, and galactose. When you combine 2 monosaccharides together, it could be the same monosaccharide or different monosaccharides, you'll produce disaccharides like maltose and sucrose and lactose. When you combine multiple units of monosaccharides together, you get polysaccharides like starch and glycogen and cellulose. Your monosaccharides typically have the same basic chemical formula, some multiple of 1 carbon to 2 hydrogen to 1 oxygen. Glucose is the most common monosaccharide that you're going to encounter. Monosaccharides can be classified based on where their carbonyl group is. Are they going to find it at the terminal end, an aldose, because it's representing an aldehyde, or are they going to find it between carbon atoms, and that would be when you have a ketose. You can have different numbers of carbons, and you can take them from being in a linear form and you can transpose them into a ring form. In general, sugars' primary purpose is that they are a fuel source for our cells, and they are also able to serve as a building block for molecules that our cells might need or organisms might need for support. So here we have glucose. You see the aldehyde at the end at carbon 1, and you see the hydroxyl group at carbon 5. When they start to circle with one another, the hydroxyl group interacts with the aldehyde group, and it causes the hydrogen that was a part of the hydroxyl to shift over to the carbonyl's oxygen. So there is not anything new formed. It's just a rearrangement of where that hydrogen is at on that molecule. So the linear form and the ring form would be structural isomers. They're the same number of carbons and hydrogens and oxygens. They're just physically connected differently. I told you that you can have sugars with different numbers of carbons. You can have trioses, 3 carbon sugars. You can have pentoses, 5 carbon sugars. And then glucose and its counterparts would be hexoses, 6 carbon sugars. Disaccharides will form when monosaccharides give up a water molecule, a hydroxyl group from one of the monosaccharides combines with a hydrogen off of another monosaccharide. That particular linkage is known as a glycosidic linkage. Depending on which carbons that linkage forms, you can have 1,4 glycosidic linkages and you can have 1,2 glycosidic linkages. Now I want you to notice with glucose how the hydroxyl group is pointing down. we're going to see that when hydroxyl groups are pointing down as opposed to pointing up, that's going to impact on how our body is able to digest some forms of sucrose of sugars, but not all. So here you see two glucoses made of maltose, and in the second example, you see a glucose and a fructose were capable of making sucrose. Polysaccharides are sugar polymers. We tend to see these more in structure and storage roles. The structure and function of your polysaccharide will depend on the types of glycosidic linkages that form, where those positions are. Is it straight? Is it branched? Here you see in starch you have both amylose and amylopectin. And amylose is just a linear chain of glucose molecules that have been combined together via glycosidic linkages. In amylopectin, we see that branching has taken place. The same for glycogen. And then where in cellulose, we just see linear chains that are connected to each other. Storage polysaccharides for plants are going to be your starch. These are alpha glucose monomers, and that's when that hydroxyl group is pointing down, that are held together by a linkage between the first and the fourth carbon. These result in helices forming. This is how your plants are able to store energy. If you have more branching take place, you can have another form of it, amylopectin. In animals, the counterpart to the one we just spoke of is glycogen. Sorry, didn't mean to jump that far ahead. So the counterpart to starch would be glycogen. This is how your body is able to store glucose on a short-term basis. It can store it in your liver. It can also store it in your skeletal muscle cells. Cellulose is a structural polysaccharide that we see in plant cells. I talked to you about that whole alpha beta thing. With cellulose, instead of using alpha glucoses, where the hydroxyl group is pointing down on that first carbon, we use beta glucose molecules where it's pointing upwards. That doesn't seem to be that big of a deal. And the biggest difference is that our body does not produce enzymes that are capable of digesting that linkage in a beta glucose molecule that they can in alpha glucose molecules. So remember when I showed you the picture of cellulose before, I'll go back to it for a second, you can see in that picture how the cellulose, the monosaccharides basically line up against one another. Since we aren't able to digest it in human food, it's going to pass straight through your digestive system. But that linkage where they're able to line up together as a unit is going to help provide a fair amount of support, which is why plants are going to be using these as their building materials. Okay, so there you see in the box at the very bottom, you can see how the O is pointing up between the two glucose molecules that are combined together. And that's the difference between beta and alpha. Not a big deal, but I did want you to know why glycogen differs from cellulose, which differs from starch. Chitin is another structural polysaccharide. It's a little bit different than what we just saw because it's got a nitrogen group attached to its base monomer. That chitin is able to provide a little bit more structure for your arthropods, and it's actually also used in surgical thread because, again, it's able to survive closing up the wound that might be needing additional time to heal up. You want a thread that's going to be durable. Our next macromolecule are going to be our lipids. Your lipids are hydrophobic in nature. Again, this is the one group that does not form as a polymer. See lots of carbons and hydrogens making up these lipids. They are going to be very hydrophobic. There are very few areas in a lipid that have an affinity for water because all they have pretty much are nonpolar covalent bonds. Although carbon and hydrogen do want electrons differently, They have relatively the same affinity for electrons. We find lipids, or the lipids that you're most likely to encounter that are going to play key roles are your fats, your phospholipids, and your steroids. Your fats are going to be consisting of glycerol and fatty acids. Glycerol consists of 3 carbons with an alcohol group. That hydroxyl group is attached to each of the carbons. And then the fatty acids are carboxyl groups that have the hydrocarbon chains attached to them. They, again, do not interact well with water because they do not have the ability to hydrogen bond nearly to the extent that water does. Yes, they have carbons and hydrogens, but remember, hydrogen bonds need to form between super electronegative atoms that have hydrogen bonded to them. The hydrocarbon chains do not have that. So water molecules are more likely to form hydrogen bonds with other water molecules and therefore not intersperse with these hydrocarbon chains. To form a fat, 3 of the fatty acids are going to join to glycerol. Each one is going to result in a dehydration where a water molecule is released, and when they do that, they form a triglyceride. So these linkages, we talked about glycosidic linkages with the carbohydrates. Here we see that we have an ester linkage, that when you lose the hydrogen from the glycerol and the hydroxyl group from that carboxyl group that was attached to the hydrocarbon chain, the fatty acid, you see the carbon and then the oxygen and then the carbonyl group. And when you see that COC double bonded O, you have an ester. You can have different types of fatty acids making up those hydrocarbon chains. You can have different lengths in terms of numbers of carbons, and you can have different types of bonds. When you have a saturated fatty acid, all of the carbons are singly bonded to one another. You have the maximum amount of hydrogen atoms you can have in that particular chain. In your unsaturated fatty acids, you have a multiple bond. You could have a double bond, you could have more than one double bond. But anytime you have one of those double bonds, you don't have the maximum amount of hydrogens that you could have, and therefore those particular fatty acids are considered to be unsaturated. And there's an example of a saturated fat as well as an example of an unsaturated fat. And I'll talk about this a little bit more on the next slide. Your saturated fats, because they are completely filled with single bonds, are able to line up basically on top of one another and solidify more readily. With your unsaturated fats, because of the multiple bond, the double bond that's present, it causes a kink. It causes bending to occur, and that makes it a lot more challenging for those fats to be able to solidify. So we typically find them in the liquid state. And that both of these saturated and unsaturated fats, when I'm talking about states, that would be at room temperature. Fats that are typically made from saturated fatty acids are an example of those would be your animal fats. Ones that are made from plants or fish tend to have your unsaturated fatty acids. You can convert those to saturated fats by adding hydrogen, also known as hydrogenation. You've probably heard this term before, hydrogenated vegetable oils. Those are a source of your trans unsaturated fats. They are not the healthiest. And it kind of comes down to just about everything in moderation. There are unsaturated fatty acids that human bodies need, and because our cells are not able to make them, or our bodies are not able to make them, we have to get them from dietary sources. Fats serve as energy storage. We can store them in adipose cells. They are also able to provide insulation, and they can help to cushion organs that we cannot live without. Another type of lipid is a phospholipid. It looks similar to the fats that we just talked about, but you will notice that there's only 2 of the hydrocarbon chains attached to the glycerol, and in place of the 3rd hydrocarbon chain, we have a phosphate group. By having the phosphate group there as opposed to the 3rd hydrocarbon chain, we have a charged group attached to it, and that's going to cause one end of this particular molecule to be hydrophilic, to be willing to interact with water, and the hydrophobic tails are still going to have that hydrophobic component. And phospholipids are what make up that cell membrane that's present in all of our cells. And because we have a bilayer of them, we see it on both sides. We see the hydrophilic heads facing both the cytoplasm, and we see them facing the extracellular fluid outside our cells. And then we see the hydrophobic tails are pointing towards each other in the middle to stay as far away from the water as they can. The other type of lipid what we're going to talk about are steroids. You see steroids when you have four carbon rings that are fused to one another. The probably the most what's a good word for it? Or their brain lapse. Probably the best known steroid would be cholesterol. We will talk a little bit more about cholesterol when we get into cell membranes. I believe that's in chapter 7. But cholesterol is definitely a key ingredient that animal cell membranes need. Again, with moderation, you don't need to have excessive amounts of cholesterol if you happen to have extremely high levels that can contribute to cardiovascular disease. Proteins. So our third macromolecule. Proteins make up the majority of the mass that's present in your cells. There are lots of different functions for proteins. We're going to touch on each of these in this next few slides. They can serve as enzymes to help to speed up reactions. They play roles with the immune system with defense proteins. They are able to serve as sources of amino acids. We see that within seeds for plants. We see that with egg whites to help nourish embryos. We see that with milk. They're able to transport proteins. Hemoglobin is able to move oxygen throughout our bodies. We also see a lot of proteins that are involved in moving substances through the cell membrane. Since you have both the hydrophilic and hydrophobic components, substances that are hydrophobic are going to need some assistance to move from the extracellular fluid into the cell membrane, or into the cytoplasm inside the cell. We have hormones like insulin. We have receptor proteins. It is thought at this time that coronavirus is able to attack cells by joining with one of the receptor proteins that is found on the outside of cells, ACE2. We also see these with nerves in order to be able to transmit chemical signals between one nerve cell and another. They're able to help with movement. We see that with both your cilia and flagella and also in your muscle cells. And they provide structure. We've got keratin, collagen, elastin. These are proteins that are found in your hair and nails, among other sources, that are able to help to provide support. Enzymes we're going to spend a fair amount of time on because enzymes are what are going to allow a lot of the metabolic pathways in our cells, or just any sort of pathway, to be able to move along a little more expeditiously. Because these enzymes can be used repeatedly, they are working quite often, and they are going to last for an extended period of time. The picture that you have there is of RNA polymerase II, and that is a key enzyme in the transcription process of DNA to mRNA. Polypeptides are going to be unbranched polymers that are made from any combination of the 20 amino acids that we have present. Proteins can be made from one polypeptide, or they can be made from multiple polypeptides, and they are folded based on their interactions among their units to produce a biologically functional molecule that is capable, like what you just saw with RNA Pol2, to be able to go out and do whatever job it needs to perform. Amino acids are going to make up these peptide chains. They will always have the same characteristics. You'll have an amino group, you'll have a carboxyl group, you'll have a carbon that joins them together. It's known as the alpha carbon. and you'll have a hydrogen on one side of the carbon. Where they're going to vary is their side chain, their R group. And depending on what is making up their side chain, they're going to have unique properties. So here at the top, you see that we have 9 amino acids that are going to be hydrophobic. And if you look at each of the R groups in yellow, you'll notice that they all consist of some combination of carbons and hydrocarbons bonded together. We have the one with methionine where you have the sulfur kind of buried in the carbons. You have the rings of carbons in phenylalanine and tryptophan. And even with proline, all of these are having lots of a nonpolar component making up their R group. That nonpolar side chain is going to be hydrophobic in nature. When we have polar side chains, which we see with the next 6 amino acids, the ones that are in that light green, we see groups that do have polar components. We see the hydroxyl groups. We see the sulfhydryl group that's separate, not buried in the hydrocarbon chain. We see amino groups that are attached to carbonyl groups. All of those are going to have an unequal sharing of electrons, which is going to allow them to be polar and more readily interact with water and therefore be hydrophilic. The remaining 5 amino acids are also hydrophilic, but they have a set charge to them. With the 2 that are in the pink, we have acidic amino acids. They produce a carboxyl group. And remember, when we have that carboxyl group, it's because we've donated proton, so that's why they would be acidic. Whereas with the 3 that are in blue, they have a terminal amino group, which is able to accept protons, which is why they're positively charged, and therefore they would be basic. So these are also polar, but because they have that net charge, they are kind of separated from just the straight polar side chains that don't have that permanent charge in the presence of water. Amino acids are going to form by peptide bonds. So we talked about how sugars form from glycosidic linkages, and we talked about how fats are going to form by making ester linkages. With amino acids, to form a peptide bond, they too are going to release a water molecule. You can have small polypeptides and you can have really large polypeptides. But regardless of what you have, you'll always have a polypeptide that has one end with the amino group, that's going to be your N terminus, and you're going to have the other end be your carboxyl group, your C terminus. Proteins have to be folded properly to be able to function appropriately, and how the protein's folded is going to play a key role in its function. We're going to talk about different aspects of the structure that have a role in that folding process. The first one is just the literal sequence of amino acids making up your protein. So that's considered to be your primary structure. And notice again with this particular protein, you still have your amino end, you still have your carboxyl end. This order of amino acids is determined from the genetic information that has been inherited, your DNA. The secondary protein structure is going to come about from those amino and carboxyl groups that are going to be present across all of the amino acids. Those are the repeating units making up your polypeptide backbone. And because there will be interactions that take place between amino acids that come across one another, you can form helices through the alpha helix, and you can form beta pleated sheets. And again, those are just going to be hydrogen bonds that are going to form between carboxyl groups and amino groups making up that backbone of your polypeptide. The tertiary protein structure is going to involve not necessarily the patterns that you saw with secondary, because it's going to be based on interactions with your R groups. And because the R groups are going to be in different locations, we're not going to see as much of that repeating pattern. We can have hydrogen bonds occur, we can have ionic bonds occur, we can have hydrophobic interactions, we can have disulfide bridges. Van der Waals interactions are just a type of hydrophobic interaction. But all of these are going to play a role in determining what that protein structure is going to be. So here as an example, between these side chain groups, we see that there are amino acids that are close enough to one another that they can form a hydrogen bond. electronegative oxygen or nitrogen can bond with a hydrogen on another amino acid side chain. We can see that there are interactions that form when you have hydrocarbon groups that are side chains get near one another. We can have ionic bonds form when you have a carboxyl group of an acidic amino acid be close to an amino group of a basic amino acid. And we can have a physical covalent bond form between side chains that both contain the sulfur atoms. So these are all going to result in folding based on where they're at in their interactions, as opposed to the repeating pattern that we see with the secondary structure. Your quaternary structure is when all the different polypeptide chains come together to form the functional biological macromolecule. One example of this is collagen. Collagen has 3 polypeptides that basically fold together like a rope. Hemoglobin has 4 different polypeptides. 2 are alpha chains and 2 are beta chains. Sickle cell disease is one example of how just minor changes in your protein structure can have really significant impacts. In this particular case, there's an amino acid that is substituted, so in your primary structure, and it causes all sorts of issues for hemoglobin and results in sickle cell disease. So you have at the amino acid 6, you have glutamine changed to valine, and because of that change, it enhances the hydrophobic nature of the beta subunit. Because of that, that's going to change interactions on the secondary level as well as on the tertiary level for that beta subunit. And when it combines with the other subunits to form the quaternary structure, rather than being their own subunit and being able to carry oxygen readily, you will see them interact more with one another and stick together, and that's going to reduce places where oxygen can be carried. As a result, your cell shape is going to change and your body's not going to be able to get the oxygen it needs as efficiently as it would with normal hemoglobin. There are other factors that can play a role with protein structure. pH, salt concentration, temperature, anything that can affect those connections that form in the secondary and tertiary structure, and that can cause it to denature. If you think about it, when you're cooking an egg, it's kind of opaque to start with, but then it gets much more cloudy as it's cooking. The heat is causing the proteins in the egg to become denatured. In order for that protein to become biologically active again, it would need to be renatured and folded back properly into the form that it needs to be in to work to perform its desired function. Protein structure is not going to be super easy to tell from just looking at its amino acids. There are various steps it has to undergo to be able to be folded properly and to make a stable structure. We'll talk a little bit more about chaperonins on the next slide, but they just kind of help the proteins be able to be folded properly. And there are some examples of diseases that truly are just a result of folding errors with proteins. So a chaperonin is a protein. It's got a hollow cylinder and it's got a cap. The unfolded polypeptide enters the chaperonin, and once it enters it, it changes the shape of the chaperonin after the cap gets put on. The environment inside becomes hydrophilic, so that's going to help to keep the hydrophilic components of your protein, get them closer to one another, and the hydrophobic components are going to draw closer to each other. And once all of that folding has taken place, the cap will come back off and the properly folded protein will be released and move on to its intended location. Excuse me. The final macromolecule we're going to talk about are nucleic acids. This is probably the one that you are the most familiar with from your first year of biology. They're able to store, transmit, and express your hereditary information. The amino acid sequence we just talked about with proteins comes from your nucleic acids. It comes from the piece of your nucleic acid that encodes it, called a gene. Your nucleic acids are made of nucleotides, lots and lots and lots of nucleotides. We can have nucleic acids exist as DNA, that's what we find in your cell's nucleus, and we can have it exist as RNA. And we can find RNA in lots of different places. So what do nucleic acids do? They're able to direct their replication. They're able to direct transcription through the synthesis of messenger RNA. And then because they're able to directly influence transcription, they're going to indirectly influence translation. When the mRNA leaves the nucleus and goes out into the cytoplasm and joins on with a ribosome that's either free in the cytoplasm or part of the rough endoplasmic reticulum, and that, along with amino acids, will give the information it needs to code for the polypeptide. Nucleic acids are also known as polynucleotides because they're made of the nucleotide monomers. Your nucleotides have a nitrogenous base. Adenine, thymine are present only for DNA. Adenine and thymine are two of the bases. Guanine, cytosine, and uracil are three others. You will only find thymine in DNA. You will only find uracil in RNA. They'll have a pentose sugar. Deoxyribose is what makes up DNA, ribose for RNA. And then they'll have one or more phosphate groups. So the nitrogenous base, the 5-carbon sugar, and the phosphate groups are what make up your nucleotides. If you don't have that phosphate group present, you have a nucleoside. So there's an example of a polynucleotide, and there's your different bases. Notice with your pyrimidines, they are single ring bases, whereas your purines are your double rings. Pyrimidines, I remember them by the CUT, CUT, cytosine, uracil, thymine, and then purines by AG, adenine and guanine. The nucleotide polymers get joined together to make a polynucleotide, and this is the one macromolecule that does not result in water forming. They actually form covalent bonds between the phosphate group on the 5-carbon of a nucleotide and the hydroxyl group on the sugar of the 3-carbon on a subsequent nucleotide. So they basically make interconnecting units of sugar and phosphate. And then the bases are going to serve as your rungs on the double helix ladder. Every sequence is going to be different based on what is present on your chromosomes. RNA molecules typically exist as just single chains, while DNAs are going to have 2 polynucleotides spiraling basically around what we call an imaginary axis. You've definitely heard the word double helix before, and they don't just code for one gene, they code for many, many, many genes. In the double helix, that 5' to 3' bond that we talked about, they run in opposite directions with one backbone running from 5' to 3 and the other one running from 3' to 5', and that's known as antiparallel. The nitrogenous bases are going to pair up so that you will always have a purine and a pyrimidine together. They do not form a covalent bond with each other. We want to be able to dislodge those bonds when we go through transcription fairly simply. But you will have hydrogen bonds hold adenine and thymine together, and guanine and cytosine in your DNA. And then in your RNA, you'll have the same thing, but instead of having thymine with adenine, you'll have uracil with adenine. Okay. So there's your DNA is your double helix. You can see the hydrogen bonds that have formed. And your transfer RNA, again, it's a single chain. It's not going to form that double helix. And so they can overlap with one another within that single chain to make more of the T shape that we see with transfer RNAs. But RNA, again, is just single-stranded. We started out in chapter 1 talking about how evolution is going to be a theme that runs throughout this whole course. DNA and proteins are one evidence of evolution because those sequences, again, are transmitted from parents to offspring, and we can see similarities in sequences with species that are more closely related than those that are further apart. So by analyzing sequences of different species, we get a better feel for how they are related on an evolutionary level. We can kind of see more as to where they fit on that tree that we saw back in chapter 1.